Next case is just how the university is dealing with this huge field of medical devices, and then with me we will have Francisco del Pozo. He's coming, and Miguel Holgado. Francisco del Pozo, he's PhD, has a PhD here. He's a full professor of the Technical University of Madrid. Has been research fellow in UK and in Imperial College in Yale, Minnesota, New York. Invited professor of several universities, and he was also the founder and director of the Center of Biomedical Technology, and he's also one of the key persons in relation to biomedical technology here. With Miguel Holgado, Miguel has a master's degree in electrical engineering here, and the doctoral degree at the Institute of Material Sciences with the CAIT, the Spanish Research Council. And he is the leader of the Optics, Photonics, and Biophotonics Technology Lab at the Center for Biomedical Technology. But he has also another profile. He's also an entrepreneur. And then after inventing, he has 5 patents. And he's also the founder of BioD Limited, focusing on the development and the commercialization of biomedical optical devices. This is one example of how to work together, the one research center at the university, and also, as a part of it, one group, and also one entrepreneur by creating a spin-off supported by the university. Then thank you very much for coming, and I don't know how you will distribute your time.
Okay, thank you, Gonzalo. Thank you, all organizers, for inviting me to give this presentation. I wouldn't say me. This is the result of many researchers. We had a lot of fun, as Glenn was saying, but also some pain. I don't know whether, Michael, you were thinking about the pain the researchers suffer, or the pain that you are trying to solve. I don't know. But nevertheless, we have some pain and a lot of fun. I'm going to talk to some very specific issue, and very important, as you really know already, is Alzheimer disease. We are trying to develop some approaches, therapeutic approaches to these. I would say also diagnosis and therapeutic approaches to face the few things that we can do with these illnesses. You know, is no cure. And the only possibility really we have is try to identify the progress of the disease as soon as possible, trying to control that progress. If we want to localize this disease, we can analyze the cognitive continuum we all have. Many people, normal people, many people develop finally Alzheimer's disease. The percentage of this evolution, it ranges from 4.2 to 6%, which is a large proportion. In the way of this continuum, we pass through in those cases that the people evolve toward Alzheimer's disease. We find two different stages, which are those that we think we have to play with. This is the MCI, mild cognitive impairment, and also subjective memory complaints. This is our experimental target. If those people have a percentage evolving to Alzheimer's disease much bigger than those, let's say, normal people, we have to try to find something here in this place to see whether something is related to those people that evolve toward a continuum. Everybody knows more or less, maybe know the numbers, but for sure the importance of the problem is that how many people is really moving and develop Alzheimer's disease in the world. To give you an example, it's not too different around the world. We have 600,000 people that develop dementia. 400,000 of them are Alzheimer's disease. The question is, are there any functional synchronization differences? of course, we place these questions once we saw some differences. So the question is, are those differences clear enough to allow us to do an early diagnosis of Alzheimer's disease? Well, that was the hypothesis we start with, and this is the kind of innovation that we have, I understand, produced. So the objective will be going from these biomarkers toward a clinical translation and finally to transfer to industry. And this is the strategy. First, we try to get some analysis of those people, MCI, the mild cognitive impairment, and MSC, is subjective memory complaints. We analyze both the behavior of those people and their memory task, and also resting, I mean, doing nothing to some extent. By doing, this analysis is performed by registering MEG, magnetoencephalography signals. What we do then, that we analyze those signals by means of some analysis tools. Then we apply some modeling of those data in order to make some classification, and finally to get some biomarkers. The biomarkers that, of course, after some clinical trial, has to be clinical individual biomarkers that should potentially be a biomarker for exploitation. I will talk about this other part today. So what we do first is then try to see if the hypothesis is try to find changes in the networks, the synchronization networks in the brain and the different tasks. Let's study those functional networks. We simply do so by the signal get this matrix. Of course, this matrix is difficult to explain or to convert in a biomarker. That's why afterward I'll show you how to convert it in a biomarker. This is the signal we obtain. Here you see in the bottom with this MEG machine. We have to develop a powerful connectivity tools that are required. By the way, this is something that has been published a couple of years ago. It's open software, and many people, by the way, is now using it. It contains a large range of different tools. And then, well, this is the kind of thing that we observe of these memory tasks, comparing controls against MCI. We say that those lines here, the whole video is much more complex, but in those lines we see difference in these two groups in the synchronization profiles, which mean, or the question is, are those changes, significant changes, really has a power to be a discriminant in the detection of these MCI situation, and then those people that evolve to AC? Well, that's the question that, of course, we saw in other places. In that case, is another ranges and regional of the brain, and this is only a conclusion that we can say that those people MCI that evolved toward AD has a significant difference between different places in the brain. Well, okay. And then if we now do the same with those people, not MCI, but also the subjective memory complaint, these are people that has no any clinical judgment about any kind of disease, but it's people that everybody knows that are complaining about how the memory is working. We again see some difference between those SMC and the control groups. Of course, to describe this mess of synchronization is not simple. We have to convert that information in simple numbers that allows us to define or to define a diagnostic criteria. Well, the obvious thing a way to analyze those things is by using the network science. And then by doing that, we will be able to distinguish one or the other. And this is the way in which we can produce the categorization of the patients. So when we are facing a problem, some people with some memory problems, we can identify clearly that they are different between those people as a collective and the control people. And then here you see some of the conclusion of the control when they evolve toward MCI, and also those MCI that evolves to 80. So apparently we are approaching to some kind of tools to do this early detection of the Alzheimer's disease. Then, in order to convert this knowledge, the findings into biomarkers, we want to compare those results with any other kind of research that we know that has some relationship with the development of Alzheimer's disease. And then I will show you very fast the results on all of these approaches. First, imaging. We have very powerful tools to identify the images. We can see many modalities of magnetic resonance as well as spectroscopy. I can tell you, and in that situation, we can observe in different places of the brain what is different in these two communities at different levels of the disease. In here we have MCI, two different kind of MCI, mild AD and moderate AD. And then the question is, well, it seems that it's also a very interesting way to approach and to do this thing. I can advance. We have failed, and that's possibly this concept of fail. As you mentioned, we fail in getting something that, apart from seeing different, to be useful to define a diagnostic criterion. Then we move another question to analyze also the tracks, the anatomical tracks, and the comparison with those functional tracks, and we also observe some kind of differences. We also compare with neuropsychology test by seeing whether our information on links and connectivity is a relationship with these neurophysiology tests. We also did something which is very clear. The real way to identify whether someone is producing or is going with a high probability to develop Alzheimer's disease is by doing a cerebrospinal fluid extraction, and then to see whether we have a protein called tau protein, and see whether there is a relationship between the tau protein and this significant connectivity aspect that we have discovered in our previous research. Also, we analyzed the relationship with some gene, specifically one which is very much related to Alzheimer's disease, and we observed also some relationship with our connectivity profiles. And then once we go those biomarkers by analyzing the connectivity of the brain through these methods, and also comparing with the result that we have with alternative, not verifiable result, we can say that we have in our hand some biomarkers. And then we start a clinical trial, multi-center clinical trial. This multi-center clinical trial, it has a very simple objective. We have to go from this group level study to an individual patient study. To do that, we run a clinical trial, and here you see the people involved in the trial. I have to say you that we were the center of this trial. So this trial, we double-blind made an experiment on this MCI, mild cognitive impairment, and tried to see whether those results we have, okay, those results we have previously can be replicated in all the sizes. So we run different time series from the MEG, those signals I showed you previously. We did it in different times. We did it in different patients. And then we defined it for all of these aspects, 6 statistical of this, and we start comparing them. I'm not going to do the details. All this data came through a blind process to ourselves, was being analyzed in the CESVIM. I don't know whether you know it. It's the supercomputer installation we have here in this campus, and it's very close to where we are now. And then, this is the process we made for analyzing the data. I'm not going to go that detail, only to give you the idea that we use many kind of classification strategies. And finally, we got this external validation process, which is extremely interesting. The number of patients we involve in this one is in the range of 125, I think, which is a good sample, but possibly not enough. And we have to continue doing that. But nevertheless, we are now, and this is the final slide, if I can make it, we are which is the situation of this work. In here, we have different, if we see here dementia and here normal, the process of someone that develop Alzheimer's disease, of course, it takes some time, but those lines want to explain how different approaches to the person that is suffering this process can be detected in time. So if we, for example, the clinical function, if we simply observe clinical function of the patient, we can see here only at the last time we can detect that this people is developing Alzheimer's disease. In other words, it's like say, well, we don't need a doctor to actually define that one. Anyone can see someone those symptoms. If you do some cognition analysis, you improve a little bit, you can do this detection a little bit earlier. This is our objective, and this is the kind of classification things that we are thinking we have in our hands. MEC-based functional connectivity. This means this is not in a scale, but I can tell you we are talking about 2.5 years earlier. If this is the case, we pretty sure that we will take the opportunity, the only opportunity I mentioned to you before, the only opportunity to control the progress of the illness, and that's all we have to say.
Very well.